On September 24, 2023, a small capsule fell through the atmosphere over the Utah desert. It was about the size of a car tire. It weighed roughly 46 kilograms, and inside it, sealed in a canister that had never been opened since it was closed in the vacuum of space, sat 121.6 grams of rock and dust scraped from the surface of an asteroid called Bennu. The capsule had travelled roughly 4 billion miles to reach that desert, not in a straight line. The spacecraft that carried it, NASA's OSIRIS-REx, had followed a looping trajectory through the inner solar system, using Earth's gravity as a slingshot to fling itself toward an asteroid that was, at the time of launch in September 2016, about 125 million miles away. The round trip took 7 years. The actual sample collection took 6 seconds. The rest was planning, flying, orbiting, mapping, and waiting. OSIRIS-REx stands for Origins, Spectral Interpretation, Resource Identification and Security, Regolith Explorer. The name is a bureaucratic construction, but the mission itself was anything but. Its purpose was to visit one of the most primitive objects in the solar system, study it from orbit, collect a sample of its surface material, and bring that sample back to Earth without exposing it to terrestrial contamination. It was the first American mission to return an asteroid sample, and only the third such mission in history, following Japan's Hayabusa and Hayabusa 2 missions. There was another reason NASA chose Bennu. The asteroid crosses Earth's orbit. In the late 21st century, roughly 150 years from now, Bennu has a small, but non-negligible chance of impacting our planet. If it did, the energy released would be equivalent to roughly 1,200 megatons of TNT, enough to devastate an area the size of a large country. Understanding Bennu's composition, its mass distribution, and how its surface interacts with sunlight, which subtly alters its orbit over time through a process called the Yarkovsky effect, was not just a matter of scientific curiosity. It was a matter of planetary defense. The same mission that returned the building blocks of life was also gathering intelligence about a potential threat to civilization. That asteroid orbits the Sun roughly 200 million miles from Earth. It is about as wide as the Empire State Building is tall, roughly 1,600 feet in diameter, and it is ancient. Bennu formed from the debris of a much larger asteroid that was itself a remnant of the earliest days of our solar system, more than 4.5 billion years ago. Before the Earth existed, before the Moon existed, before anything we would recognize as a planet had finished assembling from the disk of gas and dust that surrounded our young star. When OSIRIS-REx arrived at Bennu in December 2018, the first surprise came almost immediately. The asteroid's surface was not what anyone expected. Pre-mission models, based on telescopic observations from Earth, had predicted a relatively smooth surface with large areas of fine-grained material suitable for sampling. Instead, Bennu was covered in boulders. Rocks the size of houses littered the landscape. The smooth sandy patches that mission planners had counted on for the sampling maneuver were almost entirely absent. The team spent months recharting the surface, looking for any spot, where the sampling arm could touch down safely. They eventually identified a small clearing designated Nightingale, roughly the size of a few parking spaces, surrounded by boulders that could damage or destroy the spacecraft if the descent went even slightly wrong. NASA's OSIRIS-REx spacecraft had spent 7 years getting to Bennu, orbiting it, studying it, and then performing the single most delicate maneuver of the entire mission. On October the 20th, 2020, the spacecraft descended toward a small clearing on Bennu's surface, a spot about the size of a few parking spaces surrounded by boulders the size of buildings. It extended a robotic arm, touched the surface for approximately 6 seconds, fired a burst of nitrogen gas to stir up material, and collected what it could. The spacecraft nearly sank into the asteroid. Bennu's surface turned out to be far looser and more porous than anyone expected. The sampling arm plunged nearly half a metre into the ground before the thrusters fired to pull the spacecraft back. If the team had waited even another second, the arm might have gotten stuck. The entire collection was over in 6 seconds, but those 6 seconds gathered enough material to keep scientists busy for decades. 3 years later, the capsule carrying that material separated from the spacecraft, entered Earth's atmosphere at roughly 27,000 miles per hour, deployed a parachute over the Utah test and training range, and landed in the desert sand. Recovery teams in protective gear picked it up within minutes and transported it to a temporary clean room on site. The recovery was timed for a Sunday morning specifically to minimize the number of aircraft in the area and reduce the chance of contamination from jet exhaust or other atmospheric pollutants. Helicopters carried the capsule from the landing site to the temporary clean room in a sealed container. The container was pressurized with nitrogen to prevent any contact with the desert air. Even the dust kicked up by the helicopter rotors was a concern. The team had rehearsed the recovery dozens of times, planning for every contingency, because there would be no second chance. The sample inside the capsule was irreplaceable. From the temporary facility in Utah, the sealed capsule was flown to NASA's Johnson Space Center in Houston, Texas. There, in a dedicated curation laboratory that had been built specifically for the Bennu sample, it was opened under a constant flow of ultra-pure nitrogen gas. The laboratory had been designed with contamination control as the primary concern. The air inside was filtered to a cleanliness level far beyond what a hospital operating room requires. Every surface, every tool, every container had been cleaned, baked, and tested to ensure that no terrestrial organic molecules could interfere with the analysis of the sample. The nitrogen mattered. Meteorites that crash to Earth are contaminated the moment they touch our atmosphere. They pick up water vapor, biological material, pollutants, and trace chemicals from the environment. Even meteorites recovered within hours of landing carry the fingerprints of Earth's biosphere on their surfaces. Every amino acid, every organic molecule found in a meteorite comes with a question attached. Is this really from space, or did it come from the ground it landed on? Is this genuinely extraterrestrial, or is it a hitchhiker that climbed aboard during the rock's time on Earth? The Bennu sample had no such problem. It was collected in the vacuum of space, sealed inside a canister designed to prevent any contact with Earth's atmosphere, and opened only inside a controlled environment where the air itself had been replaced with an inert gas. For the first time in the history of American space exploration, scientists had a large, pristine sample of a carbon-rich asteroid that had never been touched by terrestrial contamination. Every molecule detected in the sample could be attributed to the asteroid with confidence, without caveats, without asterisks, without footnotes about possible contamination. When they opened the canister, the material inside was darker and more fine-grained than many had anticipated. It looked like dark powder mixed with small pebbles, some of them glossy, others rough. The team used specialized tools to handle the grains, tweezers, tiny spatulas, everything done under nitrogen, under cameras, with every motion recorded. The first challenge was simply getting the canister open. 2 of the fasteners on the TAGSAM head, the touch-and-go sample acquisition mechanism that had contacted Bennu's surface, were stuck. Engineers spent weeks developing new custom tools to remove them without disturbing the sample inside. It was a reminder that even after a flawless 7-year mission, the final steps could be the most frustrating. But by early 2024, the full sample was accessible, weighed, documented, and ready for distribution to analysis teams around the world. The total mass recovered was 121.6 grams. That is roughly the weight of a small apple. It does not sound like much, but it was far more than the mission's minimum requirement of 60 grams, and it was orders of magnitude more than what Hayabusa 2 had returned from Ryugu. The Ryugu sample was 5.4 grams. The Bennu sample was more than 22 times larger. This difference in sample size meant that Bennu could be divided among far more research teams, analysed with far more techniques, and studied for far longer than Ryugu. It also meant that most of the sample could be set aside, untouched, for future generations of scientists with future instruments and future questions. The allocation process was deliberate and careful. Small portions of the sample, typically measured in milligrams, were distributed to approved research teams at institutions across the United States, Japan, Europe, Canada, and Australia. Each team received material suited to their specific analytical techniques. Some received loose powder for chemical extraction. Others received individual grains for microscopic examination. The bulk of the sample, roughly 70 to 75%, was designated for long-term storage, sealed under nitrogen in stainless steel containers at Johnson Space Center, where it would remain available for decades to come. What they found inside that powder would challenge some of the most basic assumptions about how life's ingredients formed, where they came from, and whether they were unique to our planet. The findings arrived in waves. The first papers came in early 2025. More followed at the end of that year. And as each result was published, the picture that emerged was stranger and more significant than anyone had predicted. But to understand why the Bennu findings matter, and why they shook the fields of astrobiology and planetary science as deeply as they did, we need to go back further. Because the first space rock to rewrite biology textbooks did not arrive in a NASA capsule. It crashed through a barn roof in rural Australia in 1969, and it smelled like rotten peanut butter. The morning of September 28th, 1969, was ordinary in the small town of Murchison, Victoria, Australia. About 1,000 people lived there. The town sat along the Goulburn River in farming country, quiet and flat. Dairy farms and sheep stations stretched across the landscape. The nearest large city, Melbourne, was about 100 miles to the south. It was late September, early spring in the southern hemisphere, and the mornings were cool and still. At approximately 10:58 in the morning, a bright fireball streaked across the sky. Witnesses described it as blinding white, trailing sparks and fragments, visible in broad daylight. It broke into 3 separate pieces before vanishing, leaving behind a trail of white smoke that lingered in the air for several minutes. About 30 seconds later, a tremor shook the ground. Windows rattled, dogs barked. People who had been working outdoors stopped and looked up, trying to understand what they had just seen and heard. Fragments of the fireball had hit the earth across an area larger than 5 square miles. One piece, weighing 680 grams, punched clean through the roof of a barn and landed in a pile of hay. Residents of Murchison began collecting the fallen rocks almost immediately. Over the following days and weeks, more than 100 kilograms of fragments were recovered and sent to scientific institutions around the world. The rock was classified as a CM2, carbonaceous chondrite. That classification tells you 3 things. First, it is carbon-rich, meaning it contains significant amounts of organic material. Second, it has been altered by water at some point in its history, long before it ever reached Earth. And third, it is chemically primitive, meaning its composition has not been significantly changed by heating or geological processes since it first formed in the early solar system. Initial analyses in the early 1970s found something remarkable. The Murchison meteorite contained amino acids, not just 1 or 2 types, but a complex mixture including glycine, alanine and glutamic acid, amino acids familiar to biology, alongside unusual ones like isovaline and pseudoleucine that are rare on Earth. The researchers also noticed something important about what was absent. Serine and threonine, 2 amino acids commonly found as contaminants in terrestrial samples, were conspicuously missing. This absence was strong evidence that the amino acids in Murchison were genuinely extraterrestrial. To understand why this detection was so significant, you need to appreciate what the scientific consensus was at the time. In 1970, most scientists believed that amino acids, the building blocks of proteins, were products of biology. They were made by living cells for living cells. The idea that they could form spontaneously on a rock floating through space without any biological input whatsoever was considered possible, but not proven. The Miller-Urey experiment of 1953 had demonstrated that amino acids could form from simple gases in a laboratory, but that experiment simulated conditions on early Earth, not conditions on an asteroid. Murchison provided proof that amino acid synthesis was not limited to planets with atmospheres and lightning. It happened in space, on cold, dark, water-bearing rocks, through chemistry alone. The initial report in 1970 noted that the amino acids were racemic, meaning they existed in equal proportions of their left-handed and right-handed mirror images. This was significant. All life on Earth uses left-handed amino acids exclusively. If the amino acids in Murchison had been biological contaminants from Earth, they would have been predominantly left-handed. The fact that they were evenly split between both forms strongly suggested they had formed through chemical processes in space, not through biology. A complex mixture of hydrocarbons was also isolated from the meteorite, similar to the kinds of molecules produced in the famous Miller-Urey experiment of 1953, which demonstrated that organic compounds could form from simple gases under conditions thought to resemble early Earth. The Murchison meteorite was providing physical proof that the same chemistry could happen in space, on an asteroid, billions of years ago, but the full scope of what Murchison contained would not be understood for decades. As analytical tools improved, scientists kept returning to the meteorite and finding more. In 2010, a study using high-resolution mass spectrometry identified 14,000 distinct molecular compounds in a single sample. The researchers estimated that the meteorite might contain as many as 50,000 unique molecular compositions, with the potential for millions of distinct organic compounds in total. One rock, from one paddock in rural Australia, contained a chemical library that dwarfed anything scientists had expected to find in a space rock. In November of 2019, researchers announced the detection of ribose in the Murchison meteorite. Ribose is a 5-carbon sugar and a critical component of RNA, the molecule that many scientists believe preceded DNA as the primary carrier of genetic information in the earliest forms of life. Finding ribose in a meteorite meant that at least one of the essential building blocks of genetic material was being manufactured in space, on asteroids, before our planet even existed. The following year, NASA reported finding hexamphenyletretramine in Murchison and 2 other meteorites. This molecule can decompose in water to produce formaldehyde and ammonia, both of which are precursors to amino acids and sugars. The chemistry inside this rock was not just storing life's ingredients, it was storing the ingredients needed to make those ingredients. Then came the discovery that changed how scientists thought about the age of matter itself. Philip Heck, a curator of meteorites at the Field Museum of Natural History in Chicago, had been studying tiny grains extracted from the Murchison meteorite. These grains were not ordinary rock. They were pre-solar grains, meaning they formed before our sun was born. They are solid samples of dead stars, actual stardust trapped inside a meteorite, preserved unchanged for billions of years like insects in amber. Pre-solar grains are rare. They are found in only about 5% of meteorites that fall to Earth, and they are extraordinarily small. 100 of the largest ones would fit on the period at the end of a sentence. But the Field Museum holds the largest portion of the Murchison meteorite in the world, and the grains Heck's team studied had been extracted from fragments of the meteorite about 30 years earlier at the University of Chicago. Getting the grains out was a process. Jenica Greer, a graduate student working with Heck, described crushing fragments of the meteorite into a powder. Once all the pieces were separated, the result was a paste with what she called a pungent characteristic. It smelled, she said, like rotten peanut butter. That paste was then dissolved with acid, destroying everything that had formed in the solar system and leaving behind only the pre-solar grains that were tough enough to survive the chemical bath. Heck and his team wanted to know how old these grains were. They used a method based on cosmic ray exposure. High-energy particles fly through our galaxy constantly, and when they strike solid matter, they interact with it and create new elements. The longer a grain is exposed to cosmic rays, the more of these new elements accumulate. Heck compared it to putting a bucket out in a rainstorm. If the rain falls at a constant rate, the amount of water in the bucket tells you how long it has been sitting there. By measuring the amounts of cosmogenic neon-21 in the pre-solar grains, the team could calculate their exposure ages. Most of the grains they analysed were between 4.6 and 4.9 billion years old, slightly older than our Sun, which is 4.6 billion years old. But some of the grains were much older. The oldest ones dated to roughly 7 billion years ago, making them approximately 2.5 billion years older than the entire solar system. They were the oldest known solid material ever found on Earth, 7 billion years. To put that number in perspective, the universe itself is roughly 13.8 billion years old. These grains formed when the universe was barely half its current age, in the atmospheres of stars that lived and died long before our corner of the galaxy began to take shape. Think about what that means for a moment. You can hold this meteorite in your hand. It weighs about the same as a small watermelon. And somewhere inside it, locked in the mineral structure of grains smaller than the width of a human hair, there are fragments of stars that burned and died before our Sun, our Earth, our Moon, or any of the planets in our solar system existed. The calcium in your bones, the iron in your blood, the carbon in every cell of your body, all of these elements were forged in the cores of stars and distributed into space when those stars died. The pre-solar grains in Murchison are physical proof of that process. They are the actual debris, the literal ashes, of the stellar generation that preceded our own. Every element heavier than hydrogen and helium was created inside a star. Hydrogen and helium were produced in the first few minutes after the Big Bang, roughly 13.8 billion years ago. But carbon, nitrogen, oxygen, iron, phosphorus, all of the elements that make up rocks and water and living organisms, these were manufactured later, in the nuclear furnaces at the centres of massive stars. When those stars exhausted their fuel and exploded as supernovae, they scattered these heavier elements into the surrounding space, enriching the interstellar medium with the raw materials for new stars, new planets, and eventually new life. The pre-solar grains in Murchison are messengers from that process. Each grain carries an isotopic signature that identifies the type of star it came from. Some formed in the outflows of red giant stars, old swollen stars nearing the end of their lives. Others formed in the violent deaths of more massive stars, supernovae that burned briefly but brilliantly before detonating with the energy of billions of suns. The grains are like letters from the dead, written in the language of isotope ratios, carrying information about stars we will never see, and events that occurred in a galaxy that looked very different from the one we inhabit today. Those ancient stars exhausted their fuel, collapsed, and in their death, those expelled the particles that had formed in their outer layers. Those particles drifted through interstellar space, eventually getting swept up into the cloud of gas and dust that would become our solar system, incorporated into an asteroid, and carried to Earth inside a rock that crashed in an Australian paddock. The ages of the grains also revealed something unexpected about the history of our galaxy. Heck's team found more young grains than they expected, clustered around the 4.9 to 4.6 billion year range. Their hypothesis was that these grains formed during an episode of enhanced star formation, a sort of stellar baby boom that occurred roughly 7 billion years ago. More stars forming means more stars dying a few billion years later, which means more stardust being produced and eventually incorporated into new solar systems like ours. There was one more surprise. While examining how the minerals in the grains interacted with cosmic rays, the team discovered that pre-solar grains often travel through space, stuck together in large clusters. Heck described them as being like granola. Nobody had thought this kind of clumping was possible at such tiny scales, but the evidence was clear. The The stardust drifting between stars was not scattered as individual specks. It was travelling in groups, tiny aggregates of ancient stellar material making their way through the galaxy together. The Murchison meteorite sits in the Field Museum today, a rock from a rural Australian town that contains material older than the Sun. Amino acids that form without biology, sugars essential for genetic molecules, and the ashes of stars that died before our solar system was even a possibility. It proved conclusively that space rocks carry the raw ingredients of life and the remains of the cosmos that came before us. But in 1984, 15 years after Murchison fell, a different rock was pulled from the ice of Antarctica, and what scientists found inside would trigger a press conference at the White House, a scientific firestorm that lasted decades, and a question that remains genuinely open to this day. On December 27, 1984, a team of researchers was riding snowmobiles across the Allan Hills ice field in Antarctica. They were part of a program called ANTMET, the Antarctic Search for Meteorites, funded by the National Science Foundation. Antarctica is one of the best places on Earth to find meteorites because the dark rocks stand out against the white ice, and the slow movement of glaciers concentrates them in specific areas called blue ice fields. The team had been out for weeks, methodically scanning the ice for anything that did not belong. Roberta Score, who served as the laboratory manager of the Antarctic Meteorite Laboratory at NASA's Johnson Space Center, spotted a greenish-grey rock sitting on the ice. It weighed 1.93 kilograms, about 4 pounds. She picked it up, bagged it, and catalogued it. The rock was given the designation ALH 840-01. ALH for Allan Hills, 84 for the year, 001 for the first specimen of that season. It was placed in a collection alongside thousands of other Antarctic meteorites and shipped back to the United States. For 10 years it sat on a shelf. Nobody paid it special attention. It was just another rock in a large and growing collection. Then, in the mid-1990s, a team at Johnson Space Center took a closer look. Chemical analysis revealed something unexpected. The rock's composition was consistent with a Martian origin. It belonged to a rare class of meteorites called SNC meteorites, named after three falls, Chagouti, Nakla and Chassigny, which are all believed to have originated on Mars. But ALH 84001 did not fit neatly into any of the known SNC subgroups. It was different. It was older. The dating was extraordinary. Radiometric analysis using samarium, neodymium, rubidium, strontium, and potassium-argon methods indicated that the rock had crystallized from molten magma approximately 4.091 billion years ago. That places its formation in the earliest era of Martian history, a period when Mars is believed to have had a thicker atmosphere, a warmer surface, and liquid water flowing across its terrain. Our LH 84001 was not just a Martian meteorite. It was a Martian meteorite from the period when Mars may have been habitable. The story of how it got to Earth reads like science fiction, except every step has been verified through isotopic analysis. Roughly 17 million years ago, a large asteroid or comet struck Mars with enough force to launch fragments of the Martian crust into space. One of those fragments was this rock. It escaped Mars's gravitational pull and entered orbit around the Sun, drifting through the inner solar system for millions of years. Approximately 13,000 years ago, its orbit intersected with Earth's, and it fell through our atmosphere and landed on the Antarctic ice. In 1996, a team of NASA scientists led by David McKay, along with Everett Gibson and Kathy Thomas Keppeter, published a paper in the journal Science that made headlines around the world. They presented 4 lines of evidence suggesting that AH 84001 contained traces of ancient Martian life. The paper was the culmination of years of painstaking analysis. McKay had first become interested in the meteorite after noticing unusual features in the carbonate globules under a microscope. He brought in Thomas Keppeter, an expert in electron microscopy, to examine the structures at higher magnification. What they saw at the nanometer scale was unlike anything they had encountered in terrestrial geological samples. The features were tiny, ordered and embedded in the mineral matrix in ways that suggested they had formed in place rather than being deposited later. The team worked in relative secrecy for months, aware that any premature claim about Martian life would invite overwhelming scrutiny. They brought in additional specialists. They tested alternative explanations. They argued among themselves about the strength of the evidence, and when they finally decided to publish, they structured the paper around 4 distinct lines of evidence, reasoning that while each line might be explained away individually, the combination of all 4, found together in the same tiny region of a Martian rock, was harder to dismiss. The first line was the carbonate globules, tiny orange-brown mineral deposits found inside fractures in the rock, formed when liquid water carrying dissolved minerals seeped through the cracks. The team argued that these globules had formed at temperatures favourable for biological activity. The second line was organic molecules. The team detected polycyclic aromatic hydrocarbons, or PAHs, within the carbonate globules. On Earth, PAHs can be produced by both biological and non-biological processes, but their association with the carbonates in ALH 84001 was suggestive. The third line was the most unusual. Under a scanning electron microscope, the researchers found elongated structures within the carbonate globules that resembled tiny worm-like fossils. These structures measured between 20 and 100 nanometers in diameter. If they were fossilized microorganisms, they would be far smaller than any cellular life known at the time. But their shapes, their location within the carbonate, and their apparent integration with the surrounding mineral matrix were striking. The fourth line involved magnetite crystals. The team found tiny grains of magnetite, an iron oxide mineral, embedded in the carbonate globules. Some of these grains had a very specific size, shape and chemical purity that on Earth is associated with a particular type of organism, magnetotactic bacteria. Microbes that orient themselves using the Earth's magnetic field produce chains of chemically pure magnetite crystals to use as internal compasses. The magnetite grains in ALH 84001 were remarkably similar. The paper was published on August 16, 1996, but the news had leaked before publication. On August 7th, President Bill Clinton stood on the south lawn of the White House and addressed the nation about the potential discovery of evidence of past life on Mars. He called the finding one of the most stunning insights into our universe that science has ever uncovered. The speech was broadcast live. The next morning, newspapers around the world ran the story on their front pages. Time magazine and Newsweek both put ALH 84001 on their covers. The story dominated news cycles for weeks and sparked a surge of public interest in Mars exploration that would influence NASA's budget and mission planning for the next decade. The television show, The X-Files, featured a rock that looked suspiciously like a ALH 84001 in an episode. A Dan Brown novel incorporated a fictional version of the discovery into its plot. The meteorite had become a cultural phenomenon, and it had not even been conclusively proven to contain life. The scientific response was immediate and sharp. Critics began dismantling each of the 4 lines of evidence individually. The carbonate globules, some argued, could have formed at high temperatures through purely geological processes, not at the moderate temperatures favoured by biology. The PAH molecules were common enough that they could have been contaminants, either from Earth or from interplanetary dust. The worm-like structures were so small that many scientists questioned whether any organism could function at that scale. The minimum size for a free-living cell was thought to be significantly larger than 20 nanometers. How could anything alive fit inside a space that small? But the magnetite proved harder to dismiss. Kathy Thomas Keppeter had spent years studying the magnetite crystals in the carbonate globules. She later described her initial reaction to McKay's hypothesis with candor. She thought he was wrong. She joined the team expecting to straighten them out. Instead, what she found through electron microscopy deepened the mystery. The magnetite grains in ALH 84001 were chemically pure in a way she had never seen before in geological samples. When she searched the scientific literature, the only match for that level of purity came from biology. Magnetotactic bacteria on Earth produce magnetite with almost no chemical substitutions, no magnesium or chromium replacing the iron, because the biological processes that build the crystals are highly controlled. About 1/4 of the magnetite crystals in ALH 84001 shared these traits. Their size, shape, and composition matched what biology produces, but did not match what any known geological process could produce. A group led by D.C. Golden later demonstrated that similar features could be created in a laboratory without biological input. But McKay argued that Golden's experiments used unrealistically pure starting materials that would not exist naturally on Mars. The synthetic magnetite was a proof of concept, not a proof of what actually happened. McKay maintained that any successful abiotic explanation would need to account for all of the unusual features simultaneously, not just one at a time. The debate settled into a long stalemate. Most of the scientific community moved toward the conclusion that non-biological explanations could account for the features in ALH 84001, and the initial excitement faded from public view. But the question was never formally closed. In January of 2022, a team led by Andrew Steele at the Carnegie Institution for Science added a new chapter. Steele's group measured the hydrogen isotope ratios of the organic molecules in ALH 84001 and found that the ratio of heavy hydrogen to light hydrogen matched the values measured in Martian crustal water, not in terrestrial water. This was strong evidence that at least some of the organic molecules in the meteorite were genuinely Martian, not contamination from Earth. Steele's team also found minerals in ALH 84001 that were consistent with a geological process called serpentinization. This process occurs when water reacts with certain iron-rich and magnesium-rich minerals, producing hydrogen gas and new mineral phases. On Earth, serpentinization occurs at mid-ocean ridges and in other wet, rocky environments. It is considered one of the possible energy sources that could have powered the earliest forms of life on our planet. Finding evidence of the same process on Mars, preserved in a rock that formed 4 billion years ago, suggested that early Mars had the geochemical conditions associated with the origin of life on Earth. Steele was careful to state that this was not evidence of Martian life. It was evidence that Mars was performing the same chemistry that on our planet is linked to the conditions where life begins. The distinction matters. Having the right ingredients in the right environment does not guarantee that life emerged. But it means the possibility cannot be dismissed. The serpentinization finding opened a new door in Martian science. On Earth, serpentinization occurs in places like the Lost City hydrothermal field in the mid-Atlantic, where seawater reacts with mantle rock at temperatures between 40 and 90 degrees Celsius. These environments are home to thriving microbial communities that draw energy from the hydrogen gas produced by the chemical reactions, rather than from sunlight. Some astrobiologists consider serpentinization-driven ecosystems to be among the best analogues for the kinds of life that could exist on other planets, particularly planets and moons with subsurface water in contact with rock. If serpentinization occurred on Mars 4 billion years ago, and ALH 84001 preserves evidence of it, then early Mars had at least one of the energy sources that supports microbial life on Earth. Combined with the evidence of liquid water, moderate temperatures, and the presence of organic molecules, this paints a picture of a young Mars that was not merely potentially habitable in some abstract theoretical sense, but one that possessed specific measurable conditions known to support life on our own planet. The question, of course, is whether potential became reality. Did life actually emerge on Mars? Did it thrive for a time and then die out as the planet lost its atmosphere and its water froze or evaporated into space? Or did the chemistry come tantalizingly close without ever crossing the threshold? We cannot answer that question from a single meteorite, no matter how thoroughly we analyze it. ALH 84001 is a fragment of rock blasted off a planet drifting through space for 17 million years and landing on Antarctic ice 13,000 years ago. It is a postcard from a world that has changed dramatically since the message was written. To read the full letter, we would need to go to Mars, drill into the right rocks, and bring samples back to Earth under the same kind of controlled conditions that made the Bennu analysis so definitive. That mission, called Mars Sample Return, has been in planning for decades. Its timeline and budget remain uncertain. But the scientific case for it grows stronger with each new finding from ALH 84001, from Curiosity, and from the asteroid samples that keep demonstrating how widespread the chemistry of life is across the solar system. As of 2026, Everett Gibson, one of the original co-authors of the 1996 paper, continues to point to multiple Martian meteorites that he believes contain possible biosignatures. ALH 84001 is one. The Nakla meteorite, which fell in Egypt in 1911, is another. Yamato 000593, collected in Antarctica, is a third. Each contains features that some researchers interpret as potential traces of biological activity. The core tension remains. A quarter of the magnetite crystals in ALH 84001 have characteristics that no known inorganic process can fully replicate. The organic molecules are Martian. The geochemistry points to conditions conducive to life, and the rock itself comes from the specific period in Mars's history when liquid water is most likely to have existed on the surface. None of this proves life existed on Mars. All of it means the question is still worth asking. ALH 84001 asked a question that science cannot yet definitively answer. But while researchers spent decades debating a rock from Mars, a Japanese space agency was preparing to do something unprecedented. They were going to fly a spacecraft to an asteroid, land on its surface, collect pieces of it, and bring those pieces back to Earth, untouched and uncontaminated. And what they found in those samples would establish the baseline against which everything else, including Bennu, would be measured. The asteroid was called Ryugu. The mission was called Hayabusa 2, and the material it returned would be described as the most primitive substance in the solar system ever studied by human hands. Japan's Hayabusa 2 spacecraft launched on December 3rd, 2014, from the Tanegashima Space Center. Its target was a small, diamond-shaped, carbon-rich asteroid orbiting the Sun between Earth and Mars. The spacecraft arrived at Ryugu in June of 2018 and spent the next year and a half surveying the asteroid from orbit, mapping its surface, deploying small rovers, and preparing for 2 separate sample collection maneuvers. The first touchdown occurred on February 22nd, 2019. The spacecraft descended to the surface, made contact for a fraction of a second, fired a small projectile into the ground to kick up debris, and captured the resulting cloud of particles before ascending back to a safe distance. This collected material from Ryugu's surface. The second collection was more ambitious. In April 2019, the spacecraft fired a copper projectile at the asteroid to create an artificial impact crater, excavating material from beneath the surface. In July 2019, Hayabusa 2 descended into the newly created crater and collected subsurface material that had been shielded from billions of years of solar radiation and cosmic ray bombardment. The spacecraft departed Ryugu in November 2019 and began its long journey back to Earth. On December 6, 2020, a small re-entry capsule separated from Hayabusa 2 entered Earth's atmosphere and landed in the Australian outback near Woomera, South Australia. Inside was 5.4 grams of asteroid material. It sounds like almost nothing. But those 5.4 grams represented the first samples ever collected from a carbon-rich asteroid and returned to Earth. The samples were extracted in Japan in July of 2021. A tiny portion, at roughly 30 milligrams, was allocated to an international analysis team at NASA's Goddard Space Flight Center. The results, when they came, confirmed something that had been suspected, but never proven with such clarity. Hisayoshi Yorimoto, a geoscience professor at Hokkaido University and the leader of the initial chemical analysis team, stated it plainly. The Ryugu material, he said, is the most primitive material in the solar system we have ever studied. Its composition matched that of Sierra chondrites, a class of meteorites whose elemental ratios are the closest known match to the composition of the Sun itself. Ryugu's chemical makeup was essentially a snapshot of the raw material from which our entire solar system was built, preserved almost unchanged for 4.6 billion years. The organic analysis found 13 amino acids that could be detected and measured, with an additional 5 amino acids tentatively identified. Both protein-forming amino acids, the kind life uses to build cellular machinery, and non-protein amino acids were present. The non-protein amino acids were racemic, existing in equal proportions of their left-handed and right-handed forms. This confirmed they were formed by abiotic processes, chemical reactions occurring without biology, inside the asteroid or in the molecular cloud from which it formed. The samples also contained prebiotic organic molecules that form in the presence of liquid water, aliphatic amines, carboxylic acids, polycyclic aromatic hydrocarbons, and nitrogen-containing heterocyclic compounds were all detected. These are not exotic, obscure chemicals. They are the classes of molecules from which life's fundamental machinery is built. In 2023, uracil, one of the nucleobases used in RNA, was identified in the Ryugu samples, along with nicotinic acid, also known as vitamin B3. But the other 4 nucleobases used by life, adenine, guanine, cytosine, and thymine, were either absent or present below the limits of detection. This was a notable gap. The Murchison meteorite, which had been studied for decades, contained a wider variety of nucleobases. The question was whether the difference reflected the true chemistry of the asteroid, contamination in Murchison's record, or simply the limitations of analysing such a tiny sample. The crystals found in Ryugu contained significant amounts of water locked into their mineral structures. This meant the parent body from which Ryugu was born had formed in a cold region of the early solar system, beyond what scientists call the snow lines, the distances from the young sun where water ice and carbon dioxide ice could remain solid. The water inside the parent body had altered its chemistry roughly 5.2 million years after the solar system formed, producing minerals like dolomite, and reshaping the distribution of organic molecules within the rock. The importance of the Ryugu samples went beyond what they contained. Their importance lay in what they were not. They were not contaminated. They had never entered Earth's atmosphere. They had never sat on a laboratory shelf absorbing moisture and biological material from the air. They provided for the first time a clean baseline for understanding what a primitive, carbon-rich asteroid actually looks like, unfiltered by the messy process of crash landing on our biologically active planet. To appreciate what this means, consider the difference between studying a painting in a museum and studying a photograph of that painting taken through a dirty window. Both give you information about the painting, but only one gives you the painting itself, with all its original colours and textures intact. Meteorites are the photograph through the dirty window. They carry real information, but that information has been filtered through atmospheric entry, surface weathering, and decades or centuries of exposure to Earth's environment. Sample return missions give you the painting itself. The Ryugu samples allowed scientists to calibrate their meteorite data by comparing the chemistry of Ryugu, which is pristine, to the chemistry of Sierra chondrites like the Orgel meteorite, which fell in France in 1864 and has been stored in collections ever since. Researchers could identify exactly how terrestrial exposure had altered the meteorite's chemistry. The differences were informative. Some organic molecules in meteorites appeared to be contaminants, introduced after landing. Others were genuine extraterrestrial material that had survived the journey intact. Without the Ryugu baseline, there was no way to tell the difference with certainty. Everything scientists had learned from decades of meteorite analysis could now be checked against this pristine standard. And when the Bennu samples arrived 2 years later, they would be compared directly to Ryugu. Same type of analysis, same laboratories, same instruments. The differences between the two asteroids would turn out to be as informative as their similarities. Ryugu gave us the most primitive and uncontaminated extraterrestrial material ever examined. But it was Bennu, and the 121.6 grams, sealed in a nitrogen-filled chamber in Houston, that delivered the findings nobody was prepared for. The first results from the Bennu sample were published in late January and early February of 2025, in 2 companion papers appearing in the journals Nature and Nature Astronomy. The analyses had been conducted by teams at NASA's Goddard Space Flight Center, the Smithsonian's National Museum of Natural History, the Natural History Museum in London, and institutions across the United States, Europe, and Japan. What they found in the powder scrape from a 4.5 billion-year-old asteroid was, in several cases, unlike anything seen before in any extraterrestrial material. The amino acid results came first. Danny Glavin, a senior scientist at NASA Goddard and one of the co-lead authors of the Nature Astronomy paper, had been studying amino acids in meteorites and returned samples for years. His team identified 14 of the 20 amino acids that life on Earth uses to construct proteins. These are not rare or obscure molecules. They are the exact amino acids that human cells, plant cells, and bacterial cells all use as the fundamental building blocks of proteins, the molecular machines that power nearly every biological process on the planet. Finding amino acids in space rocks was not new. Murchison had contained them since 1970. Ryugu had contained them since 2021, but the Bennu amino acids were significant for a different reason. Because they had been collected in a sealed container in the vacuum of space and opened under nitrogen, there was no ambiguity about their origin. These amino acids were from Bennu, not from the desert where the capsule landed, not from the laboratory air, not from the hands of the scientists who handled them. They were pristine extraterrestrial organic molecules, confirmed beyond any reasonable doubt. Then came the nucleobases. The team detected all 5 of the nucleobases that life on Earth uses to store and transmit genetic information through DNA and RNA. Adenine, guanine, cytosine, thymine, and uracil. These molecules are the letters of the genetic alphabet. They are the components that, when strung together along a sugar phosphate backbone, form the double helix of DNA and the single strand of RNA. Without these 5 molecules, no living organism on Earth could replicate, grow, or pass information to its offspring. In addition to the 5 canonical nucleobases, the team also found xanthine, hypoxanthine, and nicotinic acid. Nicotinic acid is another name for vitamin B3, a molecule that plays a role in cellular metabolism in every living thing on Earth. Its presence in Bennu confirmed that the asteroid's parent body was producing not just the basic letters of the genetic code, but also accessory molecules used in the biochemical processes that support life. Japanese collaborators in the analysis, led by a team at Hokkaido University, reported that the concentration of nitrogen-containing heterocyclic compounds in the Bennu samples was approximately 5 to 10 times higher than what had been found in the Ryugu samples collected by Hayabusa 2. This difference was striking. Both asteroids are carbon-rich, both formed in the outer solar system, both experienced aqueous alteration. But Bennu's organic inventory was significantly richer than Ryugu's, suggesting that the 2 asteroids either formed in different environments, experienced different levels of water-rock interaction, or that some other variable had produced very different chemical outcomes from broadly similar starting conditions. The team also found exceptionally high abundances of ammonia in the Bennu samples. Ammonia is a simple molecule, just one nitrogen atom, bonded to 3 hydrogen atoms, but it plays a critical role in prebiotic chemistry. Under the right conditions, ammonia can react with formaldehyde, another molecule detected in the Bennu samples, to form amino acids through a process known as Strecker synthesis. The raw materials for building proteins were not only present in Bennu. The reagents needed to manufacture those raw materials from even simpler precursors were present too. In November of 2025, a subsequent paper published in the Proceedings of the National Academy of Sciences pushed the organic findings further. The research team, using both pyrolysis and wet chemistry techniques, tentatively detected trace signals of a 15th protein amino acid in the Bennu samples. That amino acid was tryptophan, a molecule that had never been found in any meteorite or any returned extraterrestrial sample before. Tryptophan is one of the rarest amino acids in biology, but it is used by living organisms on Earth to build specific proteins and to produce serotonin, a neurotransmitter. Finding it in an asteroid sample, even tentatively, expanded the known inventory of prebiotic molecules in the solar system. The same paper revealed something else of importance. Different types of rock within the Bennu sample had different organic chemistries. Some stones were richer in insoluble organic matter, others had more diverse soluble compounds. This heterogeneity indicated that the wet, alkaline environment inside Bennu's parent asteroid was not uniform. Different pockets within the rock experienced different conditions, different temperatures, different levels of water-rock interaction, and those differences produced different chemical outcomes. The asteroid's interior was not a single reaction vessel. It was more like a series of interconnected chambers, each with its own chemistry. The mineral analysis, led by Tim McCoy at the Smithsonian's National Museum of Natural History and Sarah Russell at the Natural History Museum in London, produced an equally remarkable picture. The team identified traces of 11 different minerals formed through the evaporation of water containing dissolved salts. These minerals, known collectively as evaporites, are produced when salty water slowly dries up, leaving behind crystals of the dissolved substances. On Earth, evaporites form in places like salt flats, drying lakes and shallow marine environments. What made the Bennu evaporites unusual was their completeness. The team found a sequence of minerals that recorded an evaporation process that could have lasted thousands of years or longer. This was the first time such a complete evaporite sequence had been identified in any extraterrestrial sample. Individual evaporite minerals had been found in meteorites before, but never a full progression, showing the step-by-step drying of an ancient brine. Among the minerals identified was trona, a sodium carbonate. Trona had never been found in a sample from outside Earth before. Its detection in Bennu placed the asteroid alongside only 2 other bodies in the solar system known to contain evidence of sodium carbonates and brines. One is Enceladus, a moon of Saturn whose geysers spray salt water into space. The other is Ceres, the largest object in the asteroid belt, whose surface shows deposits of sodium carbonate. Bennu, Enceladus, and Ceres now shared a chemical trait that pointed to a common process, water dissolving minerals and depositing salts, occurring in very different environments across the solar system. McCoy described the brine that once existed inside Bennu's parent asteroid in a way that made it tangible. If you had a glass of this water, he said, it would taste like salty, tepid water, not a particularly appetizing drink, but it was an environment in which prebiotic chemistry could occur. Warm, salty, minerals-rich water, the kind of setting that many origin of life researchers consider a plausible cradle for the earliest biochemical reactions. The temperature of that brine can be estimated from the minerals present. Different evaporate minerals form at different temperatures. By identifying which minerals are present and which are absent, geochemists can reconstruct the thermal history of the fluid. The Bennu evaporate sequence is consistent with a brine that existed at temperatures warm enough for complex chemistry, but not so hot as to destroy delicate organic molecules. This temperature range, roughly the range you would experience in a warm bath, overlaps with the conditions that laboratory experiments have shown to be favourable for the formation of amino acids from simpler precursors. The timeline of the brine's existence is another revealing detail. The evaporite sequence records a process that could have lasted thousands of years. This was not a flash event, not a momentary splash of water that quickly evaporated. The brines inside Bennu's Perrin asteroid persisted for an extended period, long enough for multiple generations of minerals to form as the water slowly concentrated and dried. Thousands of years of warm, salty, chemically active water. Thousands of years during which amino acids, nucleobases, and other organic molecules were bathing in a mineral-rich solution. Thousands of years during which the chemistry of life had the time and the conditions to proceed. Consider the contrast with what origin of life researchers work with in the laboratory. Many pre-Bartic chemistry experiments last hours or days. The reactions they study happen in carefully controlled flasks and beakers over short timescales. But on Bennu's Perrin asteroid, the reaction vessel was the size of a mountain. The experiment ran for millennia, and the conditions were maintained not by a scientist adjusting a thermostat, but by the natural thermodynamics of a slowly cooling asteroid body. The longest pre-Bartic experiment in history was not conducted on Earth. It was conducted inside a space rock, 4.5 billion years ago, in conditions that scientists are only now beginning to reconstruct. The team also identified phosphate minerals in the Bennu grains, specifically a vein of magnesium-sodium phosphate, roughly 1/10 of a millimetre across, formed by the evaporation of that ancient brine. Phosphorus is one of the 6 elements essential to all known life, alongside carbon, hydrogen, nitrogen, oxygen, and sulfur. It is a key component of both DNA and RNA, forming the backbone that holds the genetic code together. It is also a critical part of adenosine triphosphate, or ATP, the molecule that cells use as their primary energy currency. Every time a muscle contracts, a neuron fires, or a cell divides, ATP is involved, and the phosphorus needed for ATP was sitting in a 4.5 billion-year-old asteroid sample. By February of 2025, the picture from Bennu included amino acids, nucleobases, ammonia, formaldehyde, phosphates, and minerals formed in ancient salt water. Every category of molecule that a biochemist would list as essential for the origin of life was represented in the sample. The conditions under which those molecules had formed, warm brines rich in dissolved minerals, were exactly the kind of environment that many researchers consider favourable for pre-Bartic chemistry. But the investigators were not done. In December of 2025, 3 more papers landed simultaneously, published in Nature Geoscience and Nature Astronomy. One of them described sugars, another described a substance that had never been found in any space rock, on any asteroid, in any meteorite collection on Earth, and the third contained a finding about stardust that nobody had predicted. The sugar discovery was led by Yoshihiro Furukawa of Tohoku University in Japan. His team analysed approximately 600 milligrams of powdered Bennu material, soaking the grains in water and acid to extract any sugars, then using highly sensitive laboratory instruments to identify chemical fingerprints matching specific molecules. They found ribose, they found glucose, and they found several other sugars consistent with those involved in biological processes. Ribose is a 5-carbon sugar that forms the backbone of RNA. Every molecule of RNA in every living organism on Earth contains ribose as a structural component. Without ribose, RNA cannot exist. Without RNA, the genetic instructions that guide the construction of proteins cannot be transmitted. Ribose had been found before in 2 meteorites collected on Earth, but those detections always came with the caveat that the meteorites had been exposed to terrestrial contamination. The detection in pristine Bennu material removed that caveat entirely. Glucose was the bigger surprise. It is the primary fuel source for nearly all life on Earth. Cells break down glucose to extract energy. It is the sugar in your blood that your body monitors and regulates constantly, and it had never been identified in any extraterrestrial sample before. The Bennu detection was a first. Critically, the team did not find deoxyribose. This is the sugar that forms the backbone of DNA. Its absence was as informative as the presence of ribose. If Bennu's parent asteroid is any indication of conditions in the early solar system, then ribose, the RNA sugar, was more common than deoxyribose, the DNA sugar, in the environments where prebiotic chemistry was occurring. This finding supports a hypothesis called the RNA world, which proposes that the earliest forms of life relied on RNA as their primary molecule for both storing genetic information and catalyzing chemical reactions, with DNA evolving later as a more stable long-term storage molecule. The RNA world hypothesis has been one of the most influential ideas in origin of life research for the past 4 decades. It was first proposed in the 1960s and gained prominence in the 1980s when Thomas Seek and Sidney Altman discovered that RNA molecules can function as enzymes, catalyzing chemical reactions in the same way that proteins do. This was a breakthrough because it resolved a fundamental chicken and egg problem in biology. Proteins are needed to replicate DNA, but DNA is needed to build proteins. Which came first? If RNA can do both jobs, storing information and catalyzing reactions, then RNA could have come first, acting as both the instruction manual and the construction worker in the earliest cells. DNA and proteins could have evolved later, as specialized tools that improved on RNA's capabilities. The problem with the RNA world hypothesis has always been the question of plausibility. Can RNA form spontaneously from simple precursors under realistic prebiotic conditions? Can it assemble without enzymes? Can its components, nucleobases, ribose, and phosphate, come together in the right proportions and the right configurations without biological guidance? Bennu answered part of that question. The nucleobases are there. The ribose is there. The phosphate is there. All 3 components of RNA were found in a single pristine asteroid sample. They were not evenly distributed. They were not perfectly organized. They were scattered through the rock in different concentrations and different forms, mixed with thousands of other molecules in a chemical soup that had been sitting undisturbed for 4.5 billion years. But they were all present. The raw materials for RNA existed on asteroids before Earth formed. The RNA world did not need Earth to supply its ingredients. The solar system provided them. Furukawa put the pieces together explicitly. All 5 nucleobases, used to construct both DNA and RNA, have already been found in the Bennu samples, he noted. Phosphates have already been found. And now, the discovery of ribose means that all of the components needed to form the molecule RNA are present in Bennu. Every single part. The nucleobases to carry the genetic code, the sugars to form the backbone, the phosphate to link them together. The complete molecular toolkit for building RNA was sitting on a 4.5 billion year old asteroid. The second discovery from the December 2025 papers was stranger. A team co-led by Zach Gainsforth of the University of California, Berkeley, and Scott Sanford of NASA's Ames Research Center found a substance in the Bennu samples that had never been observed in any astromaterial. It was a polymer, a material made of long tangled chains of molecules. It was rich in nitrogen and oxygen. It had once been soft and flexible, similar in texture to used chewing gum. Exposure to radiation over billions of years had made it brittle, like a plastic lawn chair left out in the sun for too many seasons. Sanford described the substance's chemistry as resembling polyurethane, a common industrial plastic on Earth. but unlike polyurethane, which has an orderly, repeating structure, the Bennu polymer had random, hodgepodge connections, and its elemental composition varied from a particle to particle. It was not manufactured. It was assembled haphazardly by chemical reactions occurring in the cold, dark interior of a young asteroid billions of years before anything resembling a factory or a laboratory existed anywhere in the universe. Sanford called it one of the earliest alterations of materials that occurred in this rock. On this primitive asteroid that formed in the early days of the solar system, he said, we are looking at events near the beginning of the beginning. The space gum, as it came to be called in the press, appeared to have formed very early in the parent asteroid's history, possibly before there was even enough water present to drive the kinds of aqueous chemistry that produce the amino acids and nucleobases. It may represent a precursor stage of chemical complexity, a step on the ladder between simple molecules and the more organized compounds associated with biology. The implications of this are worth pausing on. If the space gum formed before the aqueous alteration phase, before the warm brines that deposited the evaporate minerals and catalyzed the formation of amino acids, then it represents a layer of chemistry that is older and more primitive than anything else in the sample. It suggests that the chemical evolution of organic matter on asteroids proceeded through stages, each building on the last. First, simple molecules condensed from the solar nebula or the interstellar medium. Then, as the asteroid's parent body warmed and water began to flow through its interior, those simple molecules were transformed into more complex ones. The space gum may be a snapshot of the intermediate stage, the point at which organic molecules had begun to link together into larger structures but had not yet been reshaped by water. If that interpretation is correct, then the Bennu sample does not just contain the products of prebiotic chemistry. It contains the process frozen at different stages, simple pre-solar molecules from before the solar system formed, intermediate polymers, like the space gum from the earliest warming of the parent body, complex amino acids, nucleobases, and sugars from the aqueous alteration phase, and evaporate minerals from the final drying of the brines. It is a chemical timeline, preserved in 121.6 grams of dark powder, spanning the full arc of asteroid evolution from raw, interstellar material to the complete molecular toolkit for life. The third paper, from December 2025, dealt with stardust. Anne Nguyen of NASA's Johnson Space Center led the analysis of pre-solar grains in the Bennu samples. Pre-solar grains, as Philip Heck's work on Murchison had established, are particles that formed in the atmospheres of dying stars before our solar system existed. They are typically rare and fragile. Many types of pre-solar grains are destroyed when asteroids experience aqueous alteration, the process of water interacting with rock that reshapes their mineral and chemical composition. Nguyen's team found that the Bennu samples contain 6 times more supernova origin dust than any other asteroid material ever studied. The abundance was unexpected. It suggested that Bennu's parent body formed in a region of the early solar nebula that was particularly enriched in the dust of dying stars. Even more surprising, some of the pre-solar grains found in Bennu were silicates, a type of pre-solar grain that is known to be easily destroyed by aqueous alteration. Their survival in the Bennu samples indicated that some portions of the asteroid's parent body had escaped the water-driven chemical processing that affected the rest of the rock. Nguyen noted that the preservation of these fragile pre-solar grains was a surprise. It illustrated that the parent body was not uniformly altered. Some material escaped the aqueous processing that reshaped the minerals and organics in other parts of the asteroid. This diversity of preservation meant the Bennu samples contained both processed material, rich in amino acids and minerals formed in salt water, and unprocessed material, carrying intact stardust from before the solar system existed. The Bennu sample, in other words, was not just a record of one era. It was a record of multiple eras. It contained material from dead stars that existed billions of years before the Sun. It contained minerals formed in warm brines on a young asteroid. It contained organic molecules assembled by chemical reactions that may have laid the groundwork for the origin of life. And it contained a mysterious polymer that formed at the very dawn of the solar system, in conditions that scientists are still working to understand. But one finding from the Bennu analyses stood apart from the rest, not because of what it revealed, but because of what it destroyed. A hypothesis about the origin of life that had been building for decades, supported by data from meteorites studied on every continent, was contradicted by the pristine evidence from a sealed canister opened under nitrogen in Houston, Texas. And the contradiction pointed to one of the oldest unsolved problems in all of biology. The problem is called chirality, and to understand why the Bennu results upended a decades-old hypothesis, you first need to understand what chirality is and why it matters to every living thing on this planet. Hold your hands out in front of you, palms facing toward you. Your left hand and your right hand are mirror images of each other. They have the same components, the same number of fingers, the same structure, but they are not identical. You cannot superimpose your left hand perfectly onto your right hand. No matter how you rotate them, the thumbs will always point in opposite directions. This property, the existence of two non-superimposable mirror images, is called chirality. It comes from the Greek word chir, meaning hand. Molecules can have chirality too. Many organic molecules, including all amino acids except glycine, exist in two mirror image forms. Scientists label these forms L and D, from the Latin laevus, meaning left, and dexter, meaning right. The L form and the D form of an amino acid contain exactly the same atoms, bonded in exactly the same way. They differ only in the three-dimensional arrangement of those atoms around a central carbon. They are molecular left hands and molecular right hands. Here is where it gets strange. All life on Earth, without exception, uses left-handed amino acids to build proteins. Every bacterium, every plant, every animal, every fungus. L amino acids only. The right-handed forms are not used. They are chemically functional. They could, in principle, build proteins, but biology has chosen one hand and one hand only. The same asymmetry applies to sugars. All life on Earth uses right-handed sugars to build DNA and RNA. The ribose in your RNA is D-ribose. The deoxyribose in your DNA is D-deoxyribose. Left-handed sugars exist, but biology ignores them. This universal preference, left-handed amino acids and right-handed sugars, is called homochirality. It is one of the defining characteristics of life on Earth, and nobody knows why it exists. The question has been debated for more than a century. In 1848, Louis Pasteur observed that tartaric acid crystals from wine vats came in two mirror image forms. He separated them by hand under a magnifying glass and discovered that each form rotated polarized light in opposite directions. This was the first identification of molecular chirality in a laboratory. Pasteur speculated that the asymmetry of life might be connected to some fundamental asymmetry in nature itself, but he could not identify what that asymmetry was. Pasteur spent years pursuing this question. He believed it was one of the deepest mysteries in science, and he was right. The asymmetry he identified in tartaric acid crystals turns out to be a universal feature of biology. Every protein in your body is built from left-handed amino acids. Every strand of DNA and RNA is built from right-handed sugars. Every cell membrane, every enzyme, every hormone follows the same rule. The consistency is absolute. There are no exceptions. In all the diversity of life on Earth, from the simplest bacterium to the most complex animal, the handedness of the molecular building blocks is always the same. The mystery is not just that life chose one hand. It is that the choice appears to be arbitrary. There is no obvious chemical reason why left-handed amino acids should work better than right-handed ones. The mirror image versions are chemically identical in isolation. They have the same stability, the same reactivity, the same energy. A protein built entirely from right-handed amino acids would fold into a mirror image of the same protein built from left-handed ones, and in principle it would function just as well. So why left? Why not right? Or why not a mixture of both? We still cannot answer this definitively, but there are several hypotheses, each with its own strengths and problems. One hypothesis involves circularly polarized light from stars. When ultraviolet light is polarized, meaning its electric field rotates in a specific direction as it travels, it can selectively destroy one mirror image of a molecule faster than the other. If the molecular clouds from which our solar system formed were bathed in circularly polarized ultraviolet light, that light could have created a slight excess of one form of amino acid over the other in the dust and gas that eventually became asteroids and planets. That slight initial bias could then have been amplified by chemical processes on early Earth until one form dominated completely. This hypothesis is appealing because circularly polarized light has actually been observed in star-forming regions of the galaxy. The Orion Nebula, one of the closest and best-studied stellar nurseries, contains regions where infrared light is circularly polarized. If similar conditions existed in the molecular cloud that formed our solar system, the light could have imprinted a slight chiral bias on the organic molecules in the pre-solar dust. That bias would have been carried into asteroids, delivered to Earth by meteorites, and amplified by terrestrial chemistry into the absolute homochirality we observe today. Another hypothesis points to the weak nuclear force, one of the four fundamental forces in physics. The weak force exhibits a phenomenon called parity violation, a slight asymmetry in how it treats matter and antimatter. This asymmetry, at the subatomic level, could in principle produce an extremely tiny energy difference between the L and D forms of a molecule. The energy difference would be fantastically small, far too small to measure directly with current instruments. But over billions of chemical reactions across billions of years, that tiny energy difference might have tilted the balance toward one form. The appeal of this hypothesis is that it connects the handedness of life to a fundamental property of the universe itself. If parity violation in the weak force favours left-handed amino acids, even by an immeasurably small amount, then the choice of handedness is not random. It is built into the laws of physics. Life anywhere in the universe, if it uses amino acids, might be expected to prefer the left-handed form. This would be a profound finding, connecting biology to particle physics in a way that no one anticipated. A third hypothesis requires no cosmic explanation at all. It proposes that the choice was random. Somewhere on early Earth, a set of chemical reactions happened to produce slightly more left-handed amino acids than right-handed ones. Perhaps a particular mineral surface catalyzed the formation of one form over the other. Perhaps a local temperature gradient or pH variation tipped the balance. Once that imbalance existed, autocatalytic feedback loops, reactions in which the product catalyzes the production of more of itself, amplified the excess until one form completely dominated. Life did not choose left because of physics or starlight. It chose left because something had to go first, and chance picked left. This hypothesis has the advantage of simplicity. It does not require any special cosmic conditions or exotic physics. It only requires that autocatalytic amplification can turn a small initial bias into a complete takeover, and laboratory experiments have demonstrated that this kind of amplification is chemically feasible. The SOI reaction, discovered in 1995, showed that certain chemical reactions can amplify a tiny initial chiral excess into near-complete homochirality in just a few cycles. If similar reactions occurred on early Earth, the choice could have been made quickly and irreversibly. Each of these hypotheses has strengths and weaknesses. None has been proven. And until recently, there was one piece of evidence that seemed to favour the cosmic explanation. Meteorites. Studies of amino acids in meteorites, going back decades, had found slight excesses of left-handed amino acids in certain samples. The Murchison meteorite, the same rock that contains 7 billion-year-old stardust and over 14,000 molecular compounds, showed a small but measurable left-handed excess in some of its amino acids. Other carbonaceous chondrites showed similar patterns. The excesses were not large, typically a few percent, but they were consistent across multiple samples and multiple laboratories. This led to an attractive hypothesis. Perhaps the bias toward left-handedness began in space, on asteroids, where circularly polarized light or some other asymmetric process created a small excess of L amino acids. When those asteroids delivered their organic cargo to early Earth through meteorite impacts, they seeded the planet with amino acids that were already slightly tilted toward the left. That built-in bias was then amplified by chemistry on Earth's surface until life emerged using left-handed amino acids exclusively. The hypothesis was elegant. It connected cosmic processes to the origin of life. It explained why the bias was universal across all life on Earth, and it was supported by the meteorite data. Then Bennu was opened. The amino acids in the Bennu samples were racemic, perfectly balanced, equal proportions of left-handed and right-handed forms with no bias, no excess, and no tilt in either direction. Jason Dworkin, the OSIRIS-REx project scientist at NASA Goddard, who co-led the amino acid analysis, was direct about the implications. The racemic result, he said, throws that theory out. The left-handed excesses found in meteorites were apparently not a feature of the original asteroid material. They were artifacts of something else. Perhaps the excesses developed during the meteorite's long exposure to the Earth's environment after landing. Perhaps they were introduced during sample handling and preparation. Perhaps the aqueous alteration inside the meteorite's parent bodies, which occurred under conditions different from those in Bennu's parent body, produced the excesses through some chemical mechanism that is not yet understood. The Ryugu samples told the same story. The non-protein amino acids in the Ryugu samples were also racemic, or very nearly so. Two independent sample return missions from two different asteroids, analysed in different laboratories using different techniques, both found the same thing. No left-handed bias. This means that whatever caused life on Earth to prefer left-handed amino acids, it did not come from space, at least not from the kind of carbonaceous asteroids that delivered organic molecules to our planet during the heavy bombardment period. The bias, wherever it originated, was terrestrial, something that happened on Earth, or to the specific material that ended up on Earth, introduced the asymmetry that would become one of the most fundamental and universal properties of life. The question then is what? What process, what event, what quirk of chemistry or physics on early Earth tipped the balance toward the left? We do not know. The chirality problem is older than the discovery of DNA. It was posed before we understood the structure of proteins. It remains unsolved in 2026, and the Bennu results have narrowed the field of possible answers by eliminating one of the most promising candidates. There is a deeper layer to this question that is worth sitting with for a moment. Chirality is not just a curiosity, it is a constraint. If you built a cell using right-handed amino acids instead of left-handed ones, the proteins it produced would be mirror images of our proteins. They would fold differently. They would interact with other molecules differently. An entire parallel biology could exist, built on the opposite hand. Some researchers have speculated about what such a biology might look like, whether it could exist alongside ours, whether life on another planet might be right-handed rather than left. If the choice of handedness was truly random, then life elsewhere in the universe might have gone the other way. An alien organism, built on the same chemical principles as terrestrial life, but with opposite chirality, would be invisible to our biology. Its amino acids would not register as food, its pathogens would not infect us, its proteins would not interact with ours. We could be standing on a planet teeming with right-handed life and never recognize it, because our tools and our assumptions are calibrated for the left. Think about what that would mean for the search for life on other worlds. When scientists design instruments to detect biosignatures, signs of life in the atmospheres or surfaces of other planets, they calibrate those instruments based on what life on Earth looks like, They look for oxygen produced by photosynthesis. They look for methane produced by microbial metabolism. They look for specific ratios of carbon isotopes that indicate biological processing. All of these signatures are based on the assumption that alien life would do chemistry in roughly the same way that terrestrial life does. But if alien life uses right-handed amino acids instead of left-handed ones, its biochemistry would be subtly different. Its enzymes would have different shapes. Its metabolic pathways would produce different byproducts. Its isotopic signatures might not match the patterns we expect. The molecules it produces would be mirror images of the molecules we produce, and many of our detection methods might not recognize them. This is not a hypothetical concern, it is an active area of research. Some astrobiologists have argued that the first thing we should look for on any potentially habitable world is not specific molecules but chiral excess, any preference for one mirror image over the other, because while the specific molecules of alien life might be unfamiliar, the property of chirality itself, the tendency to prefer one hand over the other, might be a universal signature of biology, regardless of which hand was chosen. If the Bennu results are right, and the starting materials in the solar system had no chiral preference, then the choice of handedness is a local event, something that happens on each world independently. Life on Mars, if it ever existed, might have chosen right. Life on Europa or Enceladus, if it exists in the subsurface oceans of those moons, might have chosen left or right, or might have found some other solution entirely. Each world would roll the dice independently, and each roll would produce a biology that is chemically incompatible with every other. That possibility remains speculative, but it illustrates why chirality matters. The handedness of life is not a detail, it is a door, and behind that door lies one of the most fundamental questions about whether life on Earth was inevitable given the chemistry of the universe, or whether it was an accident that could have gone differently and might have gone differently on other worlds. The Bennu samples did not answer this question, they sharpened it. They told us that the asteroids delivering organic material to early Earth were not biased. The choice happened here, under conditions we have not yet been able to reconstruct. And until we can reconstruct those conditions, the origin of Homo chirality, one of the oldest problems in biology, will remain one of the most stubborn. While astrobiologists processed the chirality results from Bennu, a rover on Mars was drilling into ancient lake sediment 4,000 miles from where ALH 84001 had originally formed, and what it found there added another layer to an investigation that was rapidly becoming one of the most complex in the history of science. NASA's Curiosity rover had been exploring Gale Crater since August of 2012. The crater is approximately 96 miles in diameter and was formed by a meteorite impact roughly 3.5 to 3.8 billion years ago. At the centre of the crater sits Mount Sharp, a layered mountain of sedimentary rock that rises about 3 miles above the crater floor. The layers of Mount Sharp record hundreds of millions of years of Martian history, laid down when the crater held a lake and rivers deposited sediment into it over geological timescales. The lake that once filled Gale Crater would have been visible from space. It was fed by rivers that cut channels through the crater rim, carrying water and sediment from the surrounding highlands into the basin below. The lake level fluctuated over time, sometimes filling a significant portion of the crater, sometimes retreating to a smaller, shallower body of water. As the lake expanded and contracted, it deposited layers of sediment, mudstone when the water was calm and deep, sandstone when rivers brought in coarser material. These layers built up over millions of years, eventually forming the lower slopes of what would become Mount Sharp. Curiosity landed at the base of that mountain and has been climbing its lower slopes ever since, drilling into rocks at various elevations to sample the chemistry of different eras in Mars's past. Each metre of elevation corresponds to a different time period, like reading a book from the bottom of the page upward. The lowest layers are the oldest, deposited when the lake was at its deepest and most stable. The higher layers record a drying Mars, a planet losing its atmosphere and its water to the cold vacuum of space. The mudstone samples from the lower slopes represent the most promising targets for organic preservation. Mudstone forms when the finest grained sediment, silt and clay, settles out of calm, standing water. On Earth, mudstones are excellent at trapping and preserving organic molecules because the tiny particles pack tightly together, creating a barrier against oxygen and other agents of chemical degradation. If organic molecules existed in the waters of Gale Crater's ancient lake, mudstone is where they would have been entombed. Curiosity carries a suite of instruments designed to analyze Martian rock and soil. The one most relevant to the organic detection is SAM, the Sample Analysis at Mars Instrument. SAM is essentially a chemistry laboratory the size of a microwave oven mounted inside the rover. It can heat rock samples to over 800 degrees Celsius, driving off volatile compounds, and then identify those compounds using a combination of gas chromatography and mass spectrometry. Gas chromatography separates molecules by their physical properties, and mass spectrometry identifies them by their mass. Together, the two techniques can identify specific organic molecules in a rock sample from millions of miles away. The limitations of SAM are important to understand. The instrument heats the sample to release its molecules, which means it can break apart larger, more complex molecules in the process. What SAM detects is not necessarily what existed intact in the rock. It may be detecting fragments of larger molecules that were torn apart by the heating. This is why the detection of decane, undecane and dodecane, which are 10, 11 and 12 carbon chains, was particularly interesting. These molecules could be fragments of even longer chain compounds, possibly fatty acids with 16, 18 or 20 carbon atoms that broke apart during the heating process. Fatty acids of those lengths are the primary structural components of cell membranes in every living organism on Earth. In March of 2025, researchers announced that Curiosity's sample analysis at Mars Instrument, known as SAM, had detected the longest organic molecules ever found on the red planet. The molecules were decane, undecane and dodecane, hydrocarbons containing 10, 11 and 12 carbon atoms respectively. These are chains of carbon and hydrogen, and on Earth molecules with these chain lengths are commonly found as fragments of fatty acids. Fatty acids are the primary structural components of cell membranes, the barriers that define the boundary between the inside of a cell and the outside world. Without cell membranes, there are no cells. Without cells, there is no life as we understand it. The detection alone was not proof of anything biological. Hydrocarbons can form through purely geological processes. Volcanic activity produces them. Meteorite impacts can synthesize them. The Fischer-Tropsch process, in which carbon monoxide and hydrogen react in the presence of a catalyst, generates chains of hydrocarbons without any biology involved. These non-biological sources are well characterized, and scientists have built models to estimate how much organic material each process could contribute to a given environment. The team, led by Alexander Pavlov at NASA's Goddard Space Flight Center, did exactly that. They modelled every known non-biological source of decane, undecane and dodecane that could plausibly have operated on ancient Mars: meteorite delivery, volcanic outgassing, Fischer-Tropsch, synthesis driven by hydrothermal activity, photochemical reactions in the ancient Martian atmosphere. Each process was quantified as carefully as the available data allowed. The result was a shortfall. The total amount of these long-chain hydrocarbons that non-biological processes could account for was less than the amount detected in the Curiosity sample. There was more organic material in the rock than non-life could explain. The paper, published in the journal Astrobiology in early 2026, stated the finding carefully. The known non-biological sources cannot fully explain the abundance of these compounds. This is not a claim of life on Mars. The researchers were explicit about that. There may be non-biological processes operating on Mars that are not yet included in the models. The geochemistry of the Martian subsurface is not well understood. Chemical reactions involving minerals, water, and carbon dioxide under conditions specific to Mars might produce long-chain hydrocarbons through pathways that have not been characterized in the laboratory. The gap between the modelled non-biological contribution and the measured abundance could be filled by a process we simply have not identified yet. But the gap exists. It is measured. It is quantified. And it resists closure by the standard set of abiotic explanations. The size of the gap matters. This is not a case where the non-biological models account for 95% of the observed organic material, and the remaining 5% might be measurement error. The shortfall is substantial enough that the team felt confident publishing it in a peer-reviewed journal and stating, in the careful language of scientific literature, that the known non-biological sources are insufficient. The phrasing in the context of astrobiology carries enormous weight. Scientists in this field are acutely aware that any claim hinting at biological processes on Mars will be scrutinized with exceptional rigor. The team would not have published the result if they were not confident that the gap was real and that their models were as complete as current knowledge allows. The finding does not stand alone. It joins a growing list of organic detections by Curiosity over the past decade. Thiophene, benzene and toluene were detected in earlier drill samples from different locations in Gale Crater. These molecules are simpler than the long-chain hydrocarbons, but their presence confirmed that organic material is preserved in Martian rocks and survives the harsh conditions of the Martian surface, including intense ultraviolet radiation, oxidizing soil chemistry, and cosmic ray bombardment. The fact that complex organic molecules can persist under these conditions tells us something about the rocks themselves. The mudstone is an effective preserver of organic chemistry, even on a planet whose surface is hostile to such molecules. The connection to ALH 84001 is hard to ignore. 30 years ago, organic molecules in a Martian meteorite launched a firestorm of debate about whether Mars had ever supported life. The debate was never resolved. The meteorite's magnetite crystals remain difficult to explain without biology. The hydrogen isotope ratios confirm the organics are Martian. The geochemistry points to serpentinization, a process linked to habitable environments. Now, on Mars itself, in ancient lake sediment dating to the era when ALH 84001 formed, Curiosity has found organic molecules in quantities that standard non-biological models cannot fully account for. The finding does not prove that life existed on Mars, but it adds to a growing body of evidence that the chemical conditions on ancient Mars were more complex, more active, and more biologically relevant than the cold, dry, dead planet we see today would suggest, and the relationship between meteorites, asteroids, and the origin of life turns out to be even more complicated than ingredients and chemistry, because there was a time in Earth's history when meteorites did not just deliver molecules, they reshaped the planet itself. And instead of destroying life, the largest impacts may have done exactly the opposite. To understand what that means, you need to picture a planet that looks nothing like the one you live on. 3,260,000,000 years ago, the Earth was a different world. If you could stand on its surface, the first thing you would notice is the sky. It was not blue. The atmosphere contained almost no free oxygen, and the gases that filled it, nitrogen, carbon dioxide, methane, water vapour, would have given the sky a hazy, possibly orange or reddish tint. There was no ozone layer, because ozone is made from oxygen, and oxygen was not yet being produced in significant quantities. Ultraviolet radiation from the sun reached the surface with an intensity that would be lethal to most modern organisms. The oceans were different too. They were rich in dissolved iron and other metals, giving the water a greenish or brownish colour rather than the blue we associate with modern seas. The iron stayed dissolved because there was no oxygen in the atmosphere to react with it and pull it out of solution. The chemistry of seawater was fundamentally different from what exists today. It was more acidic, more mineral-rich, and more chemically reactive. Life existed, but it was entirely microscopic. Single-celled organisms, bacteria and archaea, were the only inhabitants of the planet. There were no plants, no animals, no fungi, no multicellular organisms of any kind, nothing visible to the naked eye. The microbes lived in the oceans, in shallow coastal waters, in hydrothermal vents on the seafloor, and possibly in thin films on rocks exposed to the surface. They derived energy from the chemical reactions, not from photosynthesis as we know it, because the oxygen-producing form of photosynthesis had not yet evolved, or had only recently begun to appear. The continents were smaller and differently arranged than today. Plate tectonics was already operating, but the continental land masses were less extensive, and their shapes bore no resemblance to modern geography. Volcanic activity was more intense. The interior of the Earth was hotter than it is now, and heat escaping from the core drove more vigorous convection in the mantle, producing more volcanism and more frequent geological upheaval. This was the world that existed when the S2 meteorite arrived, and the planet was not done being bombarded. In the first billion years after Earth formed, impacts from asteroids and comets were frequent and severe. The late, heavy bombardment, a period of intense cratering that ended roughly 3.8 billion years ago, had left the surface scarred and molten in places. But even after that period subsided, large impacts continued to occur at irregular intervals. The inner solar system was still cluttered with debris from the formation of the planets, and that debris occasionally crossed Earth's orbit with devastating consequences. One of those impacts, roughly 3.26 billion years ago, left a signature in the rock layers of the Barberton Greenstone Belt in South Africa. This is one of the oldest geological formations on Earth, a region where ancient seafloor sediments have been preserved and uplifted over billions of years of tectonic activity. By studying the chemistry and structure of specific rock layers within the Barberton Belt, geologists can reconstruct events that occurred more than 3 billion years in the past. Nadja Drabon, an early Earth geologist and assistant professor at Harvard University's Department of Earth and Planetary Sciences, has spent years studying these ancient layers. Her focus is on a specific impact event designated S2. The meteorite responsible for the S2 impact was enormous. Estimates suggest it was up to 200 times larger than the Chicxulub impactor, the asteroid that struck the Yucatan Peninsula 66 million years ago, and triggered the mass extinction that killed the dinosaurs. The S2 object would have been roughly 50 to 200 kilometers in diameter. To visualize its size, stack 4 Mount Everests on top of each other. That gives you a rough sense of scale. The immediate effects of the S2 impact were catastrophic by any standard. The collision generated a tsunami of global proportions that swept across the oceans, ripping up the seafloor in shallow coastal zones and mixing deep ocean water with surface water. The energy released by the impact was sufficient to heat the atmosphere significantly and to boil the topmost layer of the ocean. Debris from the impact was lofted into the atmosphere and spread around the entire planet, leaving a thin layer of spherules, tiny beads of condensed rock vapor that can still be identified in the geological record today. Drabon's contribution was to look beyond the destruction. Using a methodology that involved collecting and analysing rock samples at centimetre scale intervals through the S2 impact layer, examining the sedimentology, geochemistry, and carbon isotope compositions of each sample, her team reconstructed not just the impact itself, but what happened in the days, months, and years that followed. The results, published in the Proceedings of the National Academy of Sciences in October of 2024, painted a picture that ran counter to the conventional assumption about meteorite impacts. The meteorite itself delivered phosphorus to the environment. Phosphorus, as the Bennu findings would later confirm, is one of the essential elements for life, a component of DNA, RNA, and ATP. The S2 impactor carried phosphorus from space and deposited it directly into Earth's oceans and coastal zones. The tsunami stirred the ocean from top to bottom. Deep water, rich in dissolved iron, was brought into contact with shallow water that had previously been nutrient poor. Iron is a critical element for many types of microbial metabolism. Bacteria that derive energy from oxidizing iron, converting ferrous iron to ferric iron, are among the most ancient lineages of life on Earth. By bringing iron up from the deep ocean and delivering it to shallow environments where sunlight and other energy sources were available, the tsunami effectively fertilized the coastal zones. Increased weathering and erosion of land surfaces, driven by the disruption to the atmosphere and the intense rainfall that followed the impact, washed additional nutrients from continental rocks into the ocean. These nutrients included not just iron and phosphorus, but a range of minerals and dissolved elements that microbial communities could use. Drebin's analysis of the carbon isotope signatures in the rock layers immediately above the S2 impact layer showed a distinctive shift. Iron metabolizing bacteria flourished in the aftermath of the impact. The microbial community did not merely survive the catastrophe, it thrived. The impact had created conditions, nutrient-rich, iron-saturated shallow water environments that were particularly favourable for iron metabolizing microbes. The explosion of microbial activity was temporary, lasting perhaps years to decades before conditions returned to something closer to the pre-impact state. But during that window, life responded to the destruction by expanding. Imagine the scene in the weeks and months after the S2 impact. The sky is thick with dust and condensed rock vapour. The ocean is warm, heated by the energy of the collision, and murky with stirred-up sediment. The water in the shallow coastal zones is a turbid soup of dissolved iron, phosphorus, silicates, and organic compounds, some delivered by the meteorite itself, some churned up from the deep ocean, some washed off the battered land surfaces by torrential rains. Ultraviolet light from the sun filters through the hazy atmosphere, providing energy but also posing a threat to any organism caught in the open. In this environment, iron metabolizing bacteria would have found paradise. Iron was everywhere. The energy source they depended on, the chemical reaction of converting ferrous iron to ferric iron, was virtually unlimited. The phosphorus delivered by the meteorite provided the essential nutrient for growth and reproduction. The warm temperatures accelerated metabolic rates. For these simple ancient microbes, the aftermath of a global catastrophe was the equivalent of a feast. The evidence for this microbial bloom is preserved in the rock record as a shift in carbon isotope ratios. When microbes process carbon, they preferentially incorporate the lighter isotope, carbon-12, over the heavier carbon-13. This preference leaves a signature in the carbon compounds deposited in sediment. By measuring the ratio of carbon-12 to carbon-13 in the rock layers above the S2 impact, Drebin's team could detect the fingerprint of enhanced biological activity. The signal was clear. In the decades following the impact, microbial activity increased significantly compared to the periods before and long after. Drebin's conclusion was stated with the kind of precision that distinguishes careful science from speculation. "We think of impact events as being disastrous for life," she said, "but what this study is highlighting is that these impacts would have had benefits to life, especially early on. These impacts might have actually allowed life to flourish. This finding reframes the role of meteorites in Earth's history. We tend to think of the relationship between space rocks and life in one of two ways. Either meteorites deliver the ingredients of life, amino acids, nucleobases, sugars and phosphates, to a young planet that lacked them, or meteorites destroyed life as the Chicxulub impact destroyed the dinosaurs. Drebin's work suggests a third possibility. In the earliest epochs of life on Earth, when the only organisms were simple single-celled microbes, giant impacts may have functioned as engines of biological renewal, reshuffling the chemistry of the oceans and the atmosphere in ways that opened new ecological opportunities for microbial communities. The difference between what happened 3.26 billion years ago and what happened 66 million years ago is not just the size of the impactor, it is the nature of the life that existed at the time. The Chicxulub impact killed the dinosaurs because dinosaurs were large, complex, slow-reproducing organisms that depended on stable food chains and environmental conditions. When the impact disrupted those conditions, the dinosaurs could not adapt quickly enough. They went extinct. But the microbes that inhabited Earth 3.26 billion years ago were the opposite. They were small, simple, fast-reproducing, and metabolically versatile. Many of them could switch between different energy sources depending on what was available. When the S2 impact rearranged the ocean's chemistry, delivering iron to shallow waters and phosphorus to the surface, the microbes did not collapse. They pivoted. They exploited the new conditions. They thrived in exactly the kind of disrupted, nutrient-rich environment that the impact created. This is one of the most counterintuitive findings in the history of geology. A rock the size of 4 Mount Everests slams into the planet. The ocean boils. The atmosphere superheats. Tsunamis rip across the globe. And life gets better. Not despite the catastrophe, but because of it. The impact did not just fail to destroy the biosphere. It actively promoted biological expansion by delivering nutrients and reshaping the chemistry of the ocean in ways that microbial communities were uniquely positioned to exploit. The S2 impact was not an isolated event. The geological record of the Barberton Greenstone Belt preserves evidence of multiple large impacts in the period between 3.5 and 3.2 billion years ago. At least 4 major impact events have been identified in this time window, each leaving its own layer of spherules and chemical signatures in the rock record. Each of these impacts would have produced similar effects: global tsunamis, atmospheric heating, nutrient redistribution, and each may have triggered similar blooms of microbial activity. The bombardment that we associate with destruction may have been, paradoxically, one of the forces that pushed early life toward greater diversity and resilience. Think of it like a forest fire. From a human perspective, a forest fire is a catastrophe. It destroys trees, displaces animals, and scars the landscape. But ecologically, fire is a force of renewal. It clears dead wood, releases nutrients into the soil, opens the canopy to light, and creates conditions for new growth that would not have been possible in the dense established forest. Many ecosystems depend on periodic fires to maintain their health and diversity. The early Earth may have depended on periodic impacts in a similar way. Each impact was a catastrophe at the moment of contact, but a stimulus for biological renewal in the years and decades that followed. This connection between impacts and biological renewal extends all the way back to the very origin of life. Some researchers have proposed that the energy delivered by meteorite impacts could have driven the chemical reactions necessary to assemble the first self-replicating molecules. The heat, the pressure, the rapid mixing of chemicals in impact-generated hydrothermal systems, all of these could have created conditions favourable for the origin of life itself. If this is correct, then life on Earth did not merely benefit from meteorite impacts. It may owe its existence to them. But there is an irony buried in this picture, and it connects to one of the most frustrating challenges in planetary science. The meteorites that are most likely to carry organic molecules, the carbon-rich, water-altered, chemically primitive, carbonaceous chondrites, are also the most fragile. They are the most easily destroyed by the heat of atmospheric entry. They are the most quickly degraded by exposure to Earth's weather and biosphere, and they make up a surprisingly small fraction of the meteorites that scientists actually find on the ground. Think about what happens when a small asteroid enters Earth's atmosphere. It hits the upper atmosphere at speeds between 25,000 and 160,000 miles per hour. The air in front of the object cannot get out of the way fast enough, so it compresses into a superheated shockwave. Temperatures at the surface of the incoming object can reach several thousand degrees. The outer layers of the rock melt, vaporize and stream away in a trail of hot gas and light, which is what we see as a shooting star or a fireball. For an iron meteorite, a dense sturdy chunk of metal, this process is violent but survivable. Iron meteorites are tough. They can absorb tremendous amounts of heat and mechanical stress without falling apart. They make it to the ground in one piece, or in a few large fragments, and they can sit on the surface for centuries without degrading significantly. For a carbonaceous chondrite, the experience is entirely different. These rocks are porous, fragile, and loosely held together. They contain significant amounts of volatile material, water, organic compounds, and minerals that decompose when heated. The shock wave and the heat of atmospheric entry attack them from the outside in, stripping away their most interesting chemistry layer by layer. Many carbonaceous meteoroids simply do not survive the trip. They explode in the upper atmosphere, scattering their material as fine dust that disperses into the wind and is never recovered. The ones that do survive and reach the ground are immediately under assault from a different enemy. Earth's biosphere. Within hours of landing, a meteorite begins to absorb moisture from the air. Microorganisms colonize its surface. Chemical reactions between the meteorite's minerals and terrestrial water begin to alter its composition. Every day that passes between a meteorite's fall and its recovery degrades the quality of the sample. The organic molecules that scientists are most interested in are also the most vulnerable to contamination and degradation. A study published in Nature Astronomy in 2025 examined this problem in extraordinary detail. A team led by Patrick Schober of Curtin University in Australia, working with colleagues from the Paris Observatory and institutions across Europe and Australia, analysed data from nearly 8,500 meteoroids and meteorite impacts, gathered from 19 fireball observation networks spanning 39 countries. It was the most comprehensive study of its kind, covering meteoroid entries into Earth's atmosphere from a truly global perspective. The team tracked the trajectories of incoming meteoroids, measured their speeds, calculated their orbits, and cross-referenced those orbits with the known populations of asteroids in the solar system. They then compared the types of meteoroids entering the atmosphere with the types of meteorites actually recovered on the ground. The finding was stark. Carbon-rich asteroids are abundant in the outer regions of the asteroid belt. They represent a significant fraction of the small bodies in the solar system. Spectroscopic observations from the telescope suggest that carbonaceous material is one of the most common types of asteroid surface composition in the outer belt. But carbon-rich meteorites account for less than 5% of the meteorites recovered on Earth. The ratio is wildly skewed. The reason is the filtering effect. Earth's atmosphere and the Sun's radiation act as a double filter, selectively destroying the most fragile meteoroids. The atmosphere incinerates them during entry. The Sun's heat, during the long orbital journey from the asteroid belt to Earth, bakes off volatile compounds and weakens the structural integrity of porous carbon-rich bodies. By the time they reach Earth, many have already been partially degraded, and the atmosphere finishes the job. This means that our understanding of what space rocks contain, built almost entirely on the study of meteorites recovered after falling to Earth, is biased. Fundamentally, systematically biased. We have been studying the survivors, the tough rocks that can withstand atmospheric entry and surface weathering, the fragile carbon-rich, organically rich meteoroids that represent the majority of carbonaceous material in the solar system, have been falling to Earth for billions of years, burning up in the atmosphere or disintegrating on the surface, leaving almost no trace. The implications are significant. If we only study the meteorites that survive, we are seeing a filtered, incomplete picture of what the solar system's small bodies actually contain. The carbonaceous material that burns up in the atmosphere might be even richer in organic molecules, even more diverse in its chemistry, than what we find in the survivors. We simply do not know, because we have almost never been able to study it in its original state. The Murchison meteorite was an exception, a large carbonaceous chondrite that survived entry and was recovered quickly. But even Murchison was contaminated by terrestrial exposure within hours of landing. The Bennu and Ryugu samples, collected in space and returned in sealed containers, are the only examples of truly pristine material from this class of asteroid. And the Bennu sample, with its 121.6 grams, is the largest such collection that exists. This is why sample return missions are not a luxury. They are a necessity. They are the only way to study the most common and most interesting class of asteroid material without the filters of atmospheric entry and terrestrial contamination. Everything we have learned from Bennu, every amino acid, every nucleobase, every sugar, every grain of stardust, came from a sample that bypassed those filters entirely. And every one of those findings was different, in some cases dramatically different, from what meteorite studies alone had suggested. Every finding from Bennu, the amino acids, the nucleobases, the sugars, the phosphates, the space gum, the stardust, all of it was extracted from a sample that is smaller than a bar of soap. The vast majority of the sample remains sealed in nitrogen at Johnson Space Center in Houston, waiting for future researchers with future instruments and future questions. What has been analysed so far represents a fraction of what is available. The studies published in 2025 are the beginning of a process that will continue for decades, and the picture that has emerged even from this early stage of analysis is fundamentally different from what scientists expected when the OSIRIS-REx mission was first conceived. The solar system is not a sterile place. It is saturated with the ingredients of biology. Amino acids form on asteroids in warm saltwater brines. Nucleobases assemble from simple precursors in the cold chemistry of molecular clouds and the aqueous environments of asteroid interiors. Sugars critical for RNA are manufactured in the same environments. Phosphorus, the element that links the genetic code together and powers cellular energy, is distributed through the rocky material of the inner and outer solar system. A strange polymer forms in the earliest moments of an asteroid's life, possibly representing one of the first steps in the long chemical staircase that leads from simple molecules to self-replicating systems. These ingredients are not found in one special place. They are found everywhere scientists have looked with the right tools, in meteorites from Australia, in Martian rocks from Antarctica, in samples collected from the surfaces of 2 different asteroids by 2 different space agencies, in ancient lake sediment drilled by a rover on Mars, in the geological record of impacts that reshaped Earth's chemistry more than 3 billion years ago. The chemistry of life is not rare. It is common. It is a feature of the solar system, built into the materials from which every planet, moon, asteroid and comet was assembled. The atoms of carbon and nitrogen and phosphorus in your body were forged in stars that died before the sun was born. They drifted through interstellar space, were incorporated into the disk of gas and dust that formed the solar system, condensed into asteroids, reacted with water to form amino acids and sugars, and were delivered to a young planet where, under conditions we still cannot fully reconstruct, they assembled into the first self-replicating molecules. That chain of events is not speculation. Every link in it has been observed. The pre-solar grains in Murchison are the stellar ashes. The amino acids in Bennu are the products of asteroidal chemistry. The evaporite minerals are the evidence of warm brines. The nucleobases are the letters of the genetic code, assembled in the dark interior of a space rock long before any planet was ready to use them. The fatty acid fragments on Mars are the membrane components of something that may or may not have been alive. The iron bacteria that bloomed after the S2 impact are the proof that catastrophe and creation are not opposites, they are partners. The path from stardust to life is not a straight line. It is a chain of events spread across billions of years and billions of miles, involving the deaths of ancient stars, the formation of asteroids, the chemistry of warm brines in the dark interiors of space rocks, the violence of impact on a young Earth, and a still mysterious moment when chemistry crossed the threshold into biology. Every link in that chain has now been observed, measured, and documented. The only link that remains missing is the moment of crossing itself. We know the ingredients were there. We know the conditions were favourable. We know the delivery system worked. Meteorites and asteroids raining organic material onto a planet with liquid water and an energy source. We know that life appeared on Earth within a few 100 million years of the planet becoming habitable, which is geologically fast. And we know that the same ingredients have been found on Mars, in conditions that no non-biological model can fully explain. What we do not know is how the leap happened. How did non-living chemicals become a living cell? How did a mixture of amino acids, nucleobases, sugars and phosphates floating in warm salty water organize itself into something that could reproduce, evolve, and eventually look up at the stars and wonder where it came from? This is the question that sits at the centre of everything we have discussed. It is the question that ALH 84001 hinted at in 1996. It is the question that the Murchison meteorites amino acids raised in 1970. It is the question that the Bennu sample has sharpened into the clearest focus it has ever been in. The raw materials are not the mystery anymore. The mystery is the assembly. Consider what had to happen. Individual amino acids had to link together into chains, long enough to fold into functional shapes. Nucleobases had to attach to sugars and phosphates to form nucleotides. Nucleotides had to link together into strands of RNA that could carry information and catalyze chemical reactions. A membrane had to form around a collection of these molecules, separating inside from outside, creating a boundary that defined the first cell. And all of this had to happen in an environment where the assembled structures were stable enough to persist, replicate, and begin the slow process of evolution by natural selection. Each of these steps has been demonstrated individually in the laboratory. Amino acids can polymerize under certain conditions. Nucleotides can form short RNA strands. Fatty acids can spontaneously assemble into membrane-like vesicles. But no one has ever combined all of these steps into a single continuous process that starts with simple chemicals and ends with a living cell. The gap between chemistry and biology remains the most profound gap in all of science. And yet the evidence from space rocks suggests that the universe is, in some sense, pushing toward this outcome. The chemistry that produces amino acids happens spontaneously on asteroids. The chemistry that produces nucleobases happens in molecular clouds and in warm brines on asteroid parent bodies. The chemistry that produces sugars happens in the same environments. The elements needed for membranes and energy currency are distributed throughout the rocky material of the solar system. The universe manufactures the ingredients of life as a matter of course, without instruction, without design, without intention. It does so because the laws of physics and chemistry permit it, and because the conditions that enable it are common. If the ingredients are common, and the conditions are common, and the process of assembly is the only step that remains unexplained, then the probability that assembly happened only once, only on Earth, in all those billions of years, across all those billions of asteroids, on all those potentially habitable worlds, starts to feel improbable. Not impossible, but improbable. The inference is not proof, it is a weight of evidence pushing in a direction. It is the kind of reasoning that scientists use when they cannot yet perform the definitive experiment, but can see the outlines of an answer forming in the data. The universe is full of life's ingredients. The universe is full of environments where those ingredients interact. And the universe is very, very old. 4.5 billion years on Earth alone. 13.8 billion years in total. That is a lot of time for chemistry to try things. The 121.6 grams of Bennu sample sitting in Houston will be studied for the rest of this century. New instruments will detect molecules that current technology cannot see. New questions will be asked that nobody has thought to ask yet. And the 95% of the sample that has not yet been opened will eventually yield findings that revise and extend everything discovered so far. Consider what that means in practical terms. Right now, in a clean room at Johnson Space Center, sealed containers hold portions of the Bennu sample that no scientist has ever examined. Those containers are filled with nitrogen. The seals have not been broken since the material was portioned out from the main sample. Inside those containers, amino acids, nucleobases, sugars, stardust and possibly molecules that have not yet been identified sit in the dark, unchanged, waiting. Decades from now, a graduate student who is currently in elementary school will write a proposal to NASA requesting access to a portion of the Bennu sample. They will use instruments that do not yet exist to ask questions that have not yet been formulated. They will find something that nobody alive today has imagined. And whatever they find will build on the foundation laid by the analyses of 2025. The same way those analyses built on the foundation laid by the Murchison meteorite in 1970. The same way Murchison built on the scientific tradition of studying fallen stones that stretches back centuries. This is how science works, not in dramatic leaps, usually, but in the patient accumulation of evidence over generations. Each finding adds a piece. Each piece changes the picture slightly, and over time a picture that was once completely blank fills in with detail and colour and depth until something recognisable emerges. The picture emerging from the study of space rocks is not yet complete, but it is no longer blank. We can see the outlines, we can see the ingredients, we can see the environments, we can see the timescales, we can see the processes. What we cannot yet see is the moment when all of these converge into something alive. That moment is still hidden, somewhere in the gap between chemistry and biology, somewhere in the first few 100 million years of Earth's history, in an environment we have not yet reconstructed with sufficient precision. But we are closer than we have ever been, and the distance is closing. Danny Glavin, who has spent his career studying the organic chemistry of extraterrestrial materials, said something in December of 2025 that captured the mood of the field. He said he is becoming much more optimistic that we may be able to find life beyond Earth, even in our own solar system. Not because Bennu contained life, it did not, but because Bennu contained everything life needs to start. And if those ingredients are common, if they are scattered across the solar system on every carbonaceous asteroid and in every warm, wet environment that ever existed on a rocky world, then the probability that life emerged only once, only on Earth, in all that chemistry, in all that time, across all those worlds, starts to feel very small. That is not proof. It is an inference. It is a scientist looking at the evidence and feeling the weight of probability shift. And it is where the investigation stands tonight. Out in the asteroid belt, fragments of ancient parent bodies continue to orbit the Sun, carrying their cargo of amino acids and nucleobases and sugars through the vacuum of space. They have been circling for 4.5 billion years. They will continue circling for billions more. Their chemistry is frozen in time, preserved in the cold and the dark, waiting for no one. On Mars, Curiosity's instruments continue to analyse rock samples from an ancient lake bed, measuring organic molecules that resist easy explanation. The rover has been working for more than 13 years now, far beyond its original mission plan. Its wheels are worn, its drill bits are dulled, but the data it sends back continues to surprise the scientists who study it. At Johnson Space Center, the remaining Bennu material sits sealed under nitrogen, untouched, waiting. 95% of the sample has not been analysed. It will outlast the careers of every scientist currently working on it. It will be there for the next generation, and the generation after that. In laboratories in Japan, the Ryugu samples are being studied with the same care and precision. Each new measurement adds to the baseline, sharpens the comparison with Bennu, and deepens our understanding of what carbon-rich asteroids contain and how their chemistry varies from one body to the next. And somewhere in collections around the world, pieces of the Murchison meteorite sit in glass cases in laboratory drawers. The rock that fell through a barn roof in rural Australia more than 50 years ago is still yielding new discoveries. Scientists are still finding molecules in it that they missed before. The pre-solar grains extracted from it 3 decades ago are still being analysed with newer, more sensitive techniques. The meteorite that started this entire field of investigation is not finished contributing to it. The rocks keep talking, the chemistry keeps revealing itself, and the question of whether we are alone in the universe, once the province of speculation and philosophy, is now being addressed with mass spectrometers and sealed sample canisters and the painstaking, methodical work of scientists who spend their careers measuring the handedness of molecules scraped from the surfaces of billion-year-old space rocks. We are not at the end of this story. We are, in Sanford's words, near the beginning of the beginning, and everything we find from here forward will be built on what a 121.6 gram sample of dark powder collected from an asteroid 200 million miles away revealed when it was finally opened under nitrogen in a clean room in Houston, Texas. The ingredients of life are not rare. They are everywhere. The question is no longer whether the chemistry exists. It is what happens next. And that question, the oldest and most consequential question our species has ever asked, is closer to being answered than it has ever been. But not tonight. Tonight the answers wait. The samples sit in their sealed containers, patient and undisturbed. The data sits in the papers, published and peer-reviewed, available to anyone who wants to read it. The questions sit in the minds of the people who spend their lives turning rocks into revelations, and in the minds of the students who will inherit those questions and carry them forward. The universe is 13.8 billion years old. Our solar system is 4.6 billion years old. Life on Earth is at least 3.5 billion years old, and probably older. The atoms in your body have been recycled through stars, through interstellar dust, through asteroids, through oceans, through the cells of organisms that lived and died billions of years before you were born. You are not separate from the story told by these space rocks. You are made of the same material. The carbon in your DNA was manufactured in a star. The phosphorus in your bones was distributed by a supernova. The water in your cells may have been delivered by a comet or an asteroid during the heavy bombardment that shaped the young Earth. When scientists study the Bennu sample and find amino acids and nucleobases and sugars, they are not studying something foreign. They are studying the precursors of everything alive. They are studying the backstory of biology. They are studying the first chapter of a book that eventually produced forests and coral reefs and human beings, and the curiosity that drives a scientist to spend 30 years analysing a rock the size of a golf ball pulled from Antarctic ice. And somewhere, 200 million miles from here, Bennu continues its slow orbit around the Sun, missing 121.6 grams of its surface, carrying the rest of its ancient cargo through the dark, the same way it has for 4.5 billion years, the same way it will continue to, long after the last instrument at Johnson Space Center has been powered down, and the last paper has been published, and the last scientist who touched the sample has retired. The asteroid does not know what we found inside it. It does not know what it means. It does not know that a species on a small blue planet spent years building a spacecraft, flying it across the solar system, touching its surface for 6 seconds, and carrying the collected material back home to study under nitrogen gas in a windowless room. It simply continues, patient and ancient and unaware, carrying the chemistry of stars and the raw materials of biology through a solar system that is far richer, far stranger, and far more full of possibility than anyone imagined, when a small capsule fell through the atmosphere over the Utah desert on a September morning, carrying 121.6 grams of something we are only beginning to understand. The investigation continues. The rocks keep talking. And tonight, as you listen to this, the ingredients of life drift through space on 1 billion asteroids, carried by the gravity of a star that was itself born from the ashes of stars that came before. The chain is unbroken. The chemistry is patient. And somewhere, in some lab, or on some other world, or in the warm interior of some undiscovered asteroid, the next chapter of this story is already being written. We just have not read it yet. And that is perhaps the most remarkable thing about all of this, The universe does not owe us answers. It does not owe us life. It does not owe us the chemistry that makes biology possible or the conditions that allow it to flourish, but it provides them anyway, generously, abundantly, on asteroids and in molecular clouds and in the warm brines of ancient space rocks and in the mud at the bottom of Martian craters. The ingredients are everywhere. The conditions recur. The chemistry proceeds, patient and persistent, following the laws of physics wherever they lead. Whether those laws have led to life anywhere else, on Mars or Europa or Enceladus, or on a world orbiting a star we have not yet named, remains the open question. But the evidence from space rocks, from Murchison and ALH and 84001 and Ryugu and Bennu and the ancient impact layers of South Africa, tells us that the universe is not indifferent to the possibility. It manufactures the prerequisites, it distributes them widely, and it gives them time. The rest, as they say, is chemistry, and chemistry, given enough time, has a way of surprising everyone.